So welcome everybody to Plant Science Seminar today. It's a great pleasure to introduce our speaker today, Ele Saltmarsh, who's going to be speaking to us about the use of optimization algorithms in facilitating maize adaptation to a changing climate. So I'm looking forward to hearing what you have to say, Ele.
Great, thank you. Yep, some of you may know me, you may not. I've been here 2.5 years now, but obviously most of the last year I haven't seen anyone new, so you may not have seen me. I'm in Armin Kamanian's lab. We generally work on crop ecosystem modeling. I kind of branched out a little bit from that and went into the area of optimization algorithms, and well, it's sort of like machine learning, but not quite that complex. I think it's a really interesting field. We're just about getting to the point where we're starting to merge computing sciences and biology a lot more. And looking up this sort of stuff, we've had a few papers on using optimization algorithms or other algorithms in plant biology, but not more than few. So hopefully you'll find it as interesting as I do. I'll give a brief introduction to why I'm doing this. You probably all know all of this stuff already because you work on similar areas. But the reason I was driven to work in the area of adaptation to climate change is because I grew up on a small-scale organic farm in Dorset, in the UK, obviously. We began to see changing climatic patterns. In the UK, that means even more rain than normal, but winters got less cold, less snow, and then occasionally you'd get flash freezes right as everything was starting to bud in the spring. Drought is going to be one of the worst issues worldwide. In my last few years, my undergraduate, the fields around Oxford started experiencing some really extreme drought. All the crops died and we got some massive fires. So hopefully we won't be seeing that everywhere, but it's definitely a thing that the severity of drought and the frequency of drought is going to increase in most areas worldwide. So if we look at this map, that just shows the levels of severe drought. So I think it uses the Palmer Drought Severity Index. So 3 months or more during the growing season at a drought severity index of -3. Obviously, drought is relative to the natural climate of the country. But here we can see the frequency of that sort of disastrous drought during the growing season in the baseline, which is in between 1949 and 2006. And then we get, here's some projections for 2050, and here in 2100. Obviously, it's going to increase in frequency and severity in most regions of the world. Obviously, this is very dependent on the emission scenario, as well as, you know, stuff like how we amend the soil, all the stuff that us guys are working on. But there's probably going to be quite severe drought to deal with, particularly, you know, up here in the US, around the Mediterranean, South Africa, Australia, and the South and Central Americas. So I'm working on maize. We didn't grow maize on our farm, but worldwide it's one of the most widely grown crops, and it's also one of the most susceptible to drought. So it suffers quite high levels of yield loss, even if drought is only moderate. Hopefully in the future, this work will also extend to wheat because, as you can see, it's among one of the top produced, and again suffers quite strongly under drought. But maize in particular is a very important crop to study because it is grown worldwide and it is normally cheaper than both rice and wheat. It's also grown in a lot of developing countries, or a lot of countries where they rely primarily on rain-fed agricultural systems instead of irrigated. Well, we've already seen some yield losses in maize around the world, around 3.8% on average. Lower yields have been experienced than what we would predict from a world without this sort of yield reduction. But if we look at rain-fed agricultural systems in particular, that percentage loss goes up to around 10%. So these places are also going to suffer a lot of yield losses, and they are going to suffer quite extreme drought, and they have less capacity to adapt to drought. Just really quickly, this is where maize is primarily grown around the globe. Obviously, a lot of these areas where maize is most densely grown overlap almost perfectly with the areas that are going to be most affected by drought in previous maps. If we look at the Midwest in particular, which is where I'm studying, we do hope to extend the work down to sub-Saharan Africa, maybe some other countries that rely predominantly on rain-fed systems, but being placed here, it's a perfect place to work. So if we look at maize production in the Midwest in particular, we've got 3 states. I think they're Iowa, Illinois, and Indiana, but I'm not quite used to the shapes yet. But this study looked at maize production under the best conditions, so the conditions that are shown on the maps as blue, compared to the worst conditions, which are the dark red. And the average per year for each of those conditions, and for the quintiles, the conditions in between, we see the general trend that yields are increasing year on year. But if we look at the difference between the yield of maize in the worst climatic conditions versus the best, the gap between them is getting larger over time, which we can see here more clearly in this second graph. The fraction of maize yield produced in those worst affected areas is becoming less and less over time, which suggests that sensitivity to drought is increasing. We think that's a particular problem in the Midwest just because we're growing plants at much increased densities. So that really affects their ability to cope with more severe conditions. So then on to tackling this problem. And as I said before, all of you guys work on a lot of areas around this, so you probably know a lot more than me about most of these things. I mentioned we could work on soil amendments, things like that, which could help to make the most of the water we do have, to store it before it hits the soil, maybe to stop it evaporating as much. Polycultures, a lot of people in my lab work on that. That's working on growing mixtures of crops with complementary water usage so that water use efficiency grows up. Then there's also improving the crop itself, which is what I'm going to focus on for today. Obviously, there's a lot of variation in how plants respond to drought, maybe in land races across the world, as well as well, deliberately induced variation, maybe caused by mutation, selective breeding. And we can really help to speed up adaptation to a changing climate by using that variation and introducing those traits through selective breeding or through genetic engineering. There's a big issue though in that the speed of change is unprecedented, and it typically takes, you know, at least a 10-year cycle to go one step down the line to evolve one population properly and to go test it and everything to make sure it's ready and it is successful. There are a lot of on the horizon techniques are coming up, like speed breeding, where they can pack in maybe up to 6, I believe, 6 generations into one growing year, which obviously massively speeds up the process. And things like genome selection, where instead of waiting for the crop to grow up, you can sequence its genome to decide, well, to find out whether it's got those traits you desire or not. And, you know, using these techniques, we've got down to maybe 4 years for one complete cycle, which is a massive, massive improvement. But when you consider the fact that we need production to almost double over the next 30 years now, we still want to do more, which is where my model comes in. I call it the space model because it's nice and catchy. It's basically a model of the soil plant atmosphere continuum, but it adds in an evolutionary aspect, and I'll go over what that means and why it's useful. So you're probably all familiar with the basic water balance equations. When the plant is growing, we get precipitation coming in, infiltration, runoff, drainage. The plant takes up water and transpires. And then obviously there are other aspects of plant growth, such as radiation interception and photosynthesis. So my model is a very basic model of plant growth, but it includes all of these processes going on to, so it takes in location-specific data. Right now I've got a lot of weather data from across the Midwest, and then soil data for the predominant soil type in the different locations that I've been testing. And so when the model runs, the SPAC model without the evolutionary part, the soil plant atmosphere continuum model will grow a plant based on those inputs to predict what yields you should get in those specified soil and climatic conditions. So it's pretty useful because it is so customizable. It can be adjusted through a number of different crop parameters for different crops and through the other inputs for different locations. And hopefully through using some predictions about how the climate will change, we can eventually extend this to grow plants under predicted future climates. Obviously, that's all not new. Just real quick, this is the sort of output we get. This is for Blaine County in Nebraska across a period of 5 years. And like with most other crop models, we get this gradual increase in biomass as the crop grows. It will start putting on root and shoot biomass immediately, increasing leaf biomass as time goes on, and then it'll start putting on grain biomass once it's finished flowering and reproducing. I've added here the precipitation to show you how that affects the amount of biomass accumulated. It is also a function of radiation, but it's much less susceptible to, well, no, actually, it's exactly as susceptible, but we get a lot less variation in the amount of radiation received. So here, I don't know if you can remember, but 2012 was a pretty dry year for everyone around here. And you can see that the amount of total biomass is reduced by almost a kilo per meter squared, which is a really significant reduction. So the idea of the evolutionary part of this is to change the way the crop grows by changing certain features about it, certain traits that will mean that the plant can better cope with these periods of drought. And it will be able to bring a crop that yields like this to a crop that can maybe yield a little bit more under the same conditions, like with selective breeding and all of those techniques, except it's all in silico. It's all done on the computer. The algorithm itself is very simple. I'm using an evolutionary algorithm, mostly because for me as a biologist, it's very easy to understand. But there are a lot of optimization algorithms. You've probably heard of particle swarm optimization, this stuff that mimics ants. There are a lot of ways to get to the best solution. But again, this was one of the easiest for me to understand. So what it basically does is it'll generate a population, in my case, a population of maize plants, and they all have slightly different traits. Currently, the trait we're evolving is root density, root layer density. So whether it has a lot of roots high up in the soil profile or maybe lower down, maybe in between. And it evaluates the fitness of that population using the plant growth model. It basically hands the root layer density profile over to the plant growth model and uses it as a black box model. Like that goes in, yield comes out, and we use yield as a measure of fitness, which will tell us whether we've improved on anything. We then select for the best individuals, the fittest or highest yielding individuals, cross them over. So we will pick different root layer density profiles and cross them between successful individuals. So we get some variation in the stuff we're testing and some random mutation as well. And we go back and do it all over again. And eventually we hope to see some sort of convergence on those traits or trait combinations that will give us the highest fitness value for the specified conditions. Here's a schematic that's more relevant to my own project. So if you can see here, we have what is basically the artificial equivalent of a chromosome with a load of individual genes. There don't have to be very many genes that you select on. So one option we considered initially was simply two different parameters, two different genes that controlled whether roots grew more strongly at the top or the bottom and the level of skewedness. So if we just selected on those two alone, we would only need two different genes. It'd be a very short chromosome, and it would probably be a lot quicker to find a optimum combination of those two different numbers. However, what we're doing here is dividing the soil into unique layers, so we can have maybe 4 or 5 different layers in the top 25 centimeters alone, just because we know that the soil is very strongly affected by evaporation, by different soil textures with increasing depth. And so customizing the root profile to be highly specific will allow it to, well, we hope, allow it to become much more strongly customized to that soil, so it will perform better. So currently I've been evolving on average a population of, well, a chromosome of 24 distinct genes that can be represented as a root length density profile. This one here would be a fairly strange profile. It'd have a massive increase in root density about halfway down and then again at the bottom. And that's obviously very strange compared to what we normally see. But if that did manage to somehow yield well under specific soil or climate conditions, we might pick out that chromosome, recombine it with another one to come up with a unique combination, and then maybe mutate some at random just to keep that variation going, and then go around and do it all again. I've been evolving populations for around 100 generations, and it varies in the amount of time it takes to run, but it is a matter of hours or days instead of those years I mentioned before to get hundreds of generations worth of evolution. Okay, so this is the basic root density profile you might expect to see in a typical maize crop. This is again for Blaine County in Nebraska. So as I showed you before, those cycles of increasing total biomass and total yield, they all are based on root profiles like this. So normally quite high at the top, a little bit less in the evaporative layer, and then it just decreases in density with depth. That'll get us under 2011 conditions, which were pretty dry, so not a great year. That sort of root density profile will give us a yield of 0.72 kilos per meter squared. Now, if we evolve the root length density profile, if we change the way that this profile looks, we can affect the yield. So here I have a quick output of the optimization algorithm. It's only been run for 45 generations, but I think I did 50 individuals or so. And so it's not as large as a typical population you'd see in nature, and it's not as long as we would expect natural evolution to take to reach a really good final outcome. But just for the purpose of illustrating how the algorithm works, initially the profiles that the algorithm generates can be really bad. They are not successful plants. So maybe it was initially a few profiles like this, and they absolutely tanked the crop's chances. But through successfully iterating through that evolutionary process, we see this gradual improvement year on year. And eventually we get the root density profiles converging to an optimum, which will give us the highest yield. Now, I told you before that this typical profile will give you 0.72 kilos per meter squared. And here, the optimum one we've reached is around 0.74 kilos per meter squared. Obviously, that is not much of an increase. And it does also really depend on county. So I ran Marion County in Kansas as well. And you don't get an improvement of more than 0.01 kilos per meter squared for the length of time that I've run it, which suggests maybe that the root density profiles there are already pretty well adapted to the local soils. But here we've managed to get some slight increase, and obviously I hope that being able to run more generations with more individuals will find an even bigger increase, because, you know, the more individuals we can test, and the longer we run it for, the higher the probability we come across something really good. The advantages of doing it this way, other than speed, is the amount of stuff we can experiment with. So again, I showed you the awful profile generated initially. We wouldn't see that in nature. We wouldn't be able to select on it. So on the left here, I have the profiles of just 5 individuals, the first 5 that were generated in this first generation for Blaine County. And they're all over the place. They are all quite unique, and they allow, you know, a pretty complete exploration of phenotypic space. We've got profiles that zigzag back and forth the whole way down, profiles that don't have any roots near the top, or profiles that don't have any near the bottom. We can try almost anything that you can imagine. And that's also one of the benefits of having this longer chromosome with 24 distinct genes, or here I think it's 20 distinct genes, because the soil isn't as deep as some other layers. But because we've got all of these different genes, we can get really, really specific profiles instead of just maybe one average curve. This is the profile that they were converging to. This is the one that will give you those top yields of 0.74 kilos per meter squared. It is fairly similar to our average root density profile, but we can see a few key changes. The main one is this increase in root density lower down in the soil, which is something we hypothesize will be useful in a lot of plants in drier years, obviously, because they can reach down to lower soil levels to access the soil water reserves. That's not always true though, and it is a nuanced outcome. So for example, here we see one of the individuals that underwent mutation. So if I go back again, in these last few populations, most individuals are up here at the 0.74 yield, but every so often you get an individual with a mutation. It is entirely at random which layer mutates and what it mutates to, but you can see that mutations typically cause a decrease in fitness, and that's a lot more likely as we reach the optimum, just because the more perfect you are, the easier it is to get worse instead of better. But here we have a mutation, and the main difference is, instead of having a slight increase at the bottom of the soil profile, there's this massive increase instead. Well, if I said that the roots in the bottom soil layers were really useful for getting subsoil water reserves, then why isn't this beneficial? And it does vary from case to case, but here we believe that it's because the soil is almost entirely clay. The soil will very easily store water, which is why having some roots that can access the subsoil will be able to reach a pretty consistent store of water. But it doesn't drain well either, and it easily becomes saturated. And so if you had a really high proportion of your roots down in those subsoil layers, you run the risk of having a high proportion of your roots becoming completely saturated with water, which reduces their functioning a lot. So here, during the wettest periods, this entire fraction of roots would not be able to function well at all, which is enough to reduce the yield from 0.74 kilos per meter squared to 0.68. So we can get a really quite significant change with just small changes in the density profile. This was just one individual layer that mutated with an accompanying decrease up here, because we do keep the total root length constant. So I'll go on to show you some other more interesting cases, and then I'll stop yelling at you about this. There's just really a lot of variation in the outputs of the algorithm, and it can be quite hard to find some consistent trends because so many variables affect how well or how badly a certain root profile will do. So here there are 3 other counties across the Midwest, Polk County in Minnesota, Marion in Kansas, and Clark County in Ohio. And as you can see, these are the optimum profiles, again for 2011, so a fairly dry year for the 3 different counties. And we've got Clark County, which has a fairly consistent density all the way across the profile. And then we've got Marion and Polk County showing actually relatively low root density towards the top and higher towards the bottom, which is earlier something that I told you we really don't often see in plants. And in Blaine County, this sort of profile tanked. So obviously there's something very different between them. One factor could be the soil texture. So these soils are all a lot less clay, which means that we're going to get better infiltration as well as better drainage. If I show you the precipitation for each county across the year, that could also have an effect. We can see that Marion County has fairly low rainfall overall, with a few massive events, like I don't know what this storm was here, but that is off the charts. Clark County, we've got fairly consistent high levels of precipitation. Polk County, fairly consistent, a bit higher than Marion, but it doesn't have those big rainfall events. If we tie those different factors together, then we can try to draw some predictions about why these root density profiles are acting like they do. So this is Clark County. It's a bit of a weird graph, so I apologize to you for that. But it shows the amount of precipitation that is going into runoff or evaporation or drainage. They're all shown in black because they are not accessible to the plant. And then infiltration, so water that the plant should be able to take up if it has roots where that water is located. So if you don't focus on anything in specific, but just look at the graph as a whole, we can see that there's a fair amount of blue there. A fair proportion of water is filtering in. It's not being lost to evaporation or to drainage. And that's fairly consistent across the year, which might be why for Clark County, the profile doesn't have any extreme increases in root density. The typical increase in root density towards the surface that we might see would occur maybe when there are a lot of other plants competing, and it's also competing with evaporation a lot, and it doesn't expect very frequent precipitation events. So it needs to have those roots there to intercept the water before it can be taken up by competitors. But here, because it seems to have rainfall so frequently and a good portion of it is stored, then it doesn't need that density. It's got roots to lower down as well. They can maybe continue to access water that might come around less frequently in the summer months. We see later on there's a little bit of drying, not much at all. So then we have Marion County. Marion County, Clark County, Marion County. This one was the red line on the graph, if you recall. And we see there is a fair amount of plant available water initially, but later on in summer, it does experience more severe drying. And because Marion County had lower levels of precipitation overall year round, we can see that the plant might really need to access those subsoil water reserves. There is water that is coming in that is being stored, but again, with the smaller precipitation events, slightly less frequent, and with the warmer temperatures that we would get in Kansas, the plant has to rely more heavily on these roots down in the bottom. And then Polk County in Minnesota, that one's similar in profile to Marion County somewhat, but it doesn't have quite as many roots down towards the lower layers. And that, I think, might just be a simple outcome of the slightly more frequent, slightly higher precipitation. It doesn't need to have as many roots down there relying on the subsoil water, because it is more costly to grow these deeper roots. There's just a greater carbon cost overall of going deeper into the soil, both because of the extra length and because the soil can become more challenging to grow in. And well, given that they're both fairly similar in soil texture, it's probably just the case that because this county is wetter, it doesn't need those extra roots, and so doesn't bother putting in the cost, or those that did put in the cost, that did grow those deeper roots, weren't quite as successful. So that just gives you an idea of how specific some of these predictions are, and how varied they can be from location to location. The ones I talked to you about, they're all in the Midwest. They're all, you know, close to 90 degrees and similar latitude and longitude. So the differences we see there can be quite hard to tie down, to pin on specific weather changes. If we're looking primarily at drought levels, we will be looking at precipitation mostly, some effects of temperature as well. And we did see back in Clark County, sorry, in Marion County in Kansas, temperature probably had some amount of impact on increasing evaporation, increasing competition for the water that came in. So it might be really hard to get some generalized predictions from this sort of work. But then again, we might be able to come up with something. And the benefit of doing it, like I said, in silico, is that it runs really fast and it is location specific. So maybe it doesn't entirely matter if we don't come up with some major predictions that will help to improve crops across the whole world at once. So the idea that Armin and I have been having is that potentially this could become some sort of easy to use tool for people in different locations to input their specific conditions and come out with specific predictions. And then we've still got the whole problem of producing these plants after they're evolved in silico. We've got this profile and we've probably got no plants in our population that look like that. We might have to go out and look for land races that show weird and wonderful root density profiles already. We might have to try and find the genetic basis of these the genetic control of root distribution and try to modify that. It's a very complex task. I very much doubt that there are 24 individual genes in maize that we can poke to change the root density in a specific layer of the soil. But maybe we can make some progress towards that. I'm not going to, because that is far more than a PhD's worth and I've already used up half of mine. But hopefully this sort of thing will just be able to prompt the directions that some plant breeders might go in. And maybe it can also give you guys some ideas, maybe less so about the plants themselves and more so about ways you can integrate computer science into what you're doing. After all, that is the thing I'm most excited about. I hope you guys are excited about it too, and thank you for listening.
Thank you, Ellie. We have some time for questions. If anybody has a question, please feel free to unmute yourself, or you can type in the chat box.
Yeah, I had a question for you, Ellie.
Mm-hmm.
I was wondering, how do you select, so in the beginning when you were talking about how your like optimization runs, like you have certain genes, like, and they can be just like 10 or 20 genes that get selected upon. How do you, like, are those actual genes that are curated from the maize genome that you pull that you know are related to root density, or they're just like filler and they just have some value?
Absolutely not. I don't know very much about the maize genome, and it's also very hard to relate a change in a gene to such a direct outcome in the morphology of the plant. So while it is useful to consider what the genes themselves are doing, it would become a lot harder to, as a function, to optimize. I'm already running into problems with the optimization algorithm because if you're selecting on so many different genes of so many individuals over such a long time, you're running that plant growth program I don't know, thousands of times, and my computer cannot take it. I'm trying to run it on the ICS cluster, but that's also having a little trouble. So if we had something that was even more complex, I don't know, we'd need something with a lot more capacity. It might become something that people can do in the future though, and could be a lot more useful for people who are breeding or selecting on plant genes. So it'll be an exciting prospect when it's possible.
Okay, cool. Thank you.
Ellie, can you see the chat box?
I'll bring it up, I think. Okay. So answering the question, other than planting density, what other management factors may be influencing fractional yield decline in maize in the Midwest? That's a tough question. It's probably quite variable. So the study I referenced earlier did highlight planting density because it affects the vapor pressure in the atmosphere, which can really affect transpiration. And I believe they tried to control for some factors such as changes in disease management or certain outbreaks. However, you can get, I guess, you can get management factors that interact with climate a lot. So maybe if you're already in suboptimal conditions and you don't think that your harvest is going to do well, I can imagine people investing less in fertilizer or pest treatment. Maybe that's something. But factors that I could include potentially in the crop growth model, I believe would have to be fairly limited to climatic factors that might change when the plants are closer together. Thank you.
I guess I wondered, and maybe you addressed this and I missed it, how difficult it is in your model to also modify, say, something about the anatomy of the root, like the kind of work that people have been doing here to reduce the carbon cost of the root, say, and have more cortical cells that cost the plant less to be able to grow deeper roots. Is that sort of what you were talking about? It's difficult to then simulate many, more than one trait and many evolution, generations of it?
Yeah, so yeah, exactly. The model is not very similar at all to models that the Lynch Lab has, for example. Their models are brilliantly complex, which again just increases the computational cost so much that it's very hard to include. So right now, the anatomical features of the plant are not defined. We basically see it as a block of biomass, and the only factors with some amount of detail would be the roots, the resistance in different roots in different layers of the soil, and then saturated conductivity in the soil, things like that. It's definitely a factor that I want to consider, especially if, as we predict, there is going to be a general trend for deeper roots being better, which Jonathan Lynch has talked to a lot about. And growing those deeper roots could incur a really high cost. So being able to optimize on reducing cost would be amazing. Hopefully we'll be able to do it sometime. The water table definitely is a factor, I think, that influences root growth and architecture. The root density profile I showed you for Blaine County in Nebraska, that had a fairly shallow soil already. So whereas most of the soils I tested run down to 2 meters deep, Blaine County only went down to 1.5 meters. And then in the optimized profile, we saw it had the majority of its roots up in the top half of that already. And again, that is probably just because the water table does fill up a lot more easily there. And so having those deeper roots would not be beneficial. In the other paper that I've seen that did use genetic algorithms, that worked on several trees growing in the Netherlands, and they generated optimum profiles for a number of different trees and then compared them to what they saw in these trees in real life. And one of the major differences was a tree that they predicted would have quite dense roots lower down really did not. It was confined almost entirely to the shallowest layer. And that again was just because the water table filled up so strongly over winter that the roots were actually a disadvantaging factor.
I guess your diagrams of the how much water infiltrated depending on the soil texture also got me thinking that management practices that prevent soil compaction or improve water infiltration and some of the structural benefits that can come with more roots in the system with cover crops, etc., could almost be included there in accounting for improving water infiltration. It just sort of got me thinking about, oh yeah, with a model you could again simulate the value.
Exactly. That's an awesome thing. I'm glad you picked up on that, because obviously growing these roots in the system has a regenerative aspect to it, and improving water quality, soil quality will feed back massively on the amount of roots that can grow there and how well the roots can do, how well the soil stores water and stuff like that. So right now we're looking at optimizing water uptake. In the future, I really do think there is the potential to extend this to select for other factors. So fitness here is a function of yield. If we looked at optimizing roots for nitrogen uptake, again, we could look at yield, but then if we wanted to optimize plants for regenerative properties, we could look at improvements in soil texture or something like that and optimize on that. And hopefully that wouldn't be massively more computationally expensive, because again, we could simply act on something like root length density profiles like I did here. We can extend it to other aspects, such as how aggressive the roots push through soil and stuff. Again, fingers crossed we'll be able to do that when the computers get better.
Great, thanks. Thank you.
Thank you.
Thank you, Ellie. Any other questions? Well, let's all thank Ellie for a wonderful presentation. Thank you very much.
Thank you.